As evolutionary biologists, we are fascinated by convergence, the independent evolution of similar phenotypes in two or more lineages. Convergent adaptation demonstrates the impressive ability of selection to repeatedly shape phenotypes in response to ecological challenges. It can also be used as a tool to study adaptation, as we get to see the process happen multiple times independently. The level of convergence can often be quite precise, with selection acting on the same genetic mechanism. For example, in these distinct vertebrate lineages, we see the adaptation towards reduced pigmentation on their light colored substrates using the same gene. These sorts of examples, maybe I wasn't talking to the mic the entire time. These sorts of examples give us insight into how repeatable evolution can be and possible constraints on evolutionary change. Okay, so when we see these sorts of genotypic convergent adaptation, there's multiple ways of which this can occur. So if you imagine that we have four populations related to each other in this tree structure here, and populations 2 and 3 are in some novel selective environment that is the same, there are three ways in which this can happen. First, there could be independent mutations at the same locus. So populations 2 and 3 have an independent mutation once they've been isolated from each other. Alternatively, migration can move the beneficial allele between selected populations. So here it's originating in population 2 and migrating into population 3. And lastly, selection can be acting on the same shared ancestral variation that's arising in the ancestor of the two selected populations. Distinguishing between these modes gives us insight into larger questions in evolutionary biology, as how adaptation is happening, whether it's occurring with standing variation or mutation, what's the role of gene flow, and other insights into these constraints. And if we look at this across populations, we can use population genetic tools to distinguish between these modes. So what I'm going to present today is a way to model neutral allele frequencies around repeatedly selected sites and put this into a likelihood framework to distinguish between these modes. But before I incorporate selection, I need to set up how neutral allele frequencies look under a model of drift alone. So going back to our four populations, now at a single neutral locus, we have present-day allele frequencies x1 through x4, and they're derived from some ancestral population where the allele frequency is epsilon. To describe this neutral population structure, we can set up a matrix of coancestry coefficients that describe the amount of drift within and between population pairs. So for example, the coancestry coefficient within the first population, population 1, is F11, and this describes the amount of drift within the single first population. This also has other connotations that you're likely to be familiar with, such as the inbreeding coefficient and FST for the first population relative to the ancestral population. Another way to think about this is the probability that a pair of lineages sampled from the first population coalesce before they reach the ancestral population. We can also define coancestry coefficients between populations. So here for populations 1 and 2, we have F12, and also the same interpretation of the probability of lineages, this time one sample from population 1, the other from population 2, coalescing before they reach the ancestral population. In this tree, populations 3 and 4 also share some coancestry, but no other pairs of populations do, just because they split after the ancestral population. Incorporating selection into this, we know that selection is changing neutral allele frequencies if they're near selected sites. And so what I'm going to do is define what these matrices of coancestry coefficients look like under various models of convergent adaptation, using this coalescent definition of pairs of lineages coalescing. And I'm just going to walk through these kind of quickly so you can get a flavor of what these models look like. In the independent mutation case, where populations 2 and 3 are interselection and the beneficial alleles rising on the same gene independently, what we see now is that there's increased coancestry within selected populations. And this is changing as we move away from the selected site. So what I'm plotting here is the coancestry coefficient against the recombination distance from the selected site, which is occurring all the way on the left. And what we see is immediately near the selected site, these neutral alleles are being forced to coalesce during the sweep. But as we move further away, there's some probability that they'll recombine off the beneficial background, and it's decreasing the probability of coalescing. And I should say that throughout the talk, when I show plots like this, the solid lines are theoretical expectations, and the dashed line is simulations that I've done. Also in this independent mutation model, we see no increased coancestry between selected populations because the sweep is independent in the two populations. In the other two models, where migration or standing variation is occurring, we again see increased coancestry within selected populations. However, now we also see increased coancestry between selected populations because the sweep is shared, whether it's via migration or selection acting on the same shared standing variation. And this is causing an additional probability of coalescing between the two selected populations. The way that we can distinguish between these modes is that the rate of decay of this coancestry is changing depending on which mode is happening. So particularly in the standing variation case, as the variant has been standing for longer in the two populations once they've been isolated from each other, there's more chance for recombination to occur, which is decreasing the probability of coalescing rapidly as you move away from the selected site. Additionally, there's some information about the direction of migration in our migration model. I don't really have time to go into the details of what's happening here, but we see increased coancestry between the recipient population and any population that shares drift with the source. So here, populations 1 and 3 and population 2 is the source. Okay, so we can define models that describe this coancestry matrix for these various modes of convergent adaptation, and they're for a particular site a given distance away from a selected site. So if you imagine that we have this black X where a selected site is, and the red dot where we're looking a certain distance away. And we can model these neutral allele frequencies of multivariate normal around our ancestral mean epsilon with variance covariance proportional to this coancestry matrix under these various models. And with that, we can calculate probabilities of our likelihood a given distance away from our selected site under a given model of convergent adaptation. And we can do this all given distances for all of our data, moving away from both sides of the selected site. And take the product of all these likelihoods to get a composite likelihood of our entire observed data for a region. Okay, so I have done just that, and I've applied this method to this really interesting case of convergent adaptation, where we see populations of killifish on the east coast of the US responding rapidly to industrial pollutant toxicity in the water. And this work is done in collaboration with Noah Reed and Andrew Whitehead, who have sampled 4 population pairs of polluted and reference populations along their range. And in particular, what I'm going to show you today is how we've applied our method to a region that we think is interesting. So we already think that genotypic convergence adaptation is happening because we see increased FST between all pairs of polluted and reference populations. And what I'm going to show you is calculating the likelihood of this allele frequency data in this region under 5 distinct models of genotypic convergence. And so these models are the straightforward ones that I laid out earlier, where all populations have independent mutations once they've been isolated from each other, where all populations share the same standing ancestral variation that selection is acting upon, or where all populations share the same variant via migration. Alternatively, because of what we know about the system and that the 2 populations in the south form a distinct clade relative to the 3 polluted populations in the north, I'm going to test the hypotheses that there's something different happening in the north relative to the southern site. So that there's standing variation in the north and an independent sweep in the south, or there's migration in the north and an independent sweep in the south. And so to orient you to these results, what I'm plotting is the composite log likelihood of a given convergent adaptation model relative to our neutral model where nil selection is happening for any proposed selected site. And what this is able to do is, 1, identify the loci involved in convergent adaptation, because these likelihoods should peak as you're moving the proposed selected site to a correct selected site. And we're doing this in a region where we know that something might be happening, but you can imagine doing this across your entire genome and looking for regions that are particularly interesting. And we're also able to distinguish between modes because at the most likely selected site, there can be a model that has the highest likelihood. So again, for a given proposed selected site, what you'll see is 5 points that correspond to these 5 convergent adaptation models. And these are the results. So there's 2 things to notice. First of all is that all of our models are peaking at the most likely proposed selected site. This is likely where the selection is happening. And this corresponds to where we see FST peaking. And if you want to know more about the genes involved in this and more to this convergence story, Andrew's talking tomorrow at 1:15. Additionally, what we see is that the models where something different is happening in the north relative to the south are most likely models. They best fit our observed data. And given this, we can also generate parameter estimates under our models. So what we see is that our selection coefficient is peaking at about 15%, which is quite strong, but we know this adaptation has been recent and rapid. And additionally, when we look at our standing variation in the north and independent sweep in the south model, what we see is that the minimum age of the standing variant, so how long our beneficial allele has been standing in each population independently, is peaking at 0 generation. So this implies that either the variant is very recently standing or migration is actually pushing it between these populations in the north. Okay, so to conclude, I can make models of these distinct modes of convergent adaptation and put this into a statistical framework that can both identify the loci involved in convergence and distinguish between these modes. And I've shown that in killifish, the adaptation towards pollutant tolerance seems to be happening via two distinct modes, migration and an independent sweep in the southern population pair. And lastly, what I want to emphasize is that because of the way these models are generated, we can do this for multiple populations with any arbitrary relationships to each other. And you can imagine many scenarios in which you can do this under, including having different populations be the source of migration, or different pairs of populations doing different things. And with that, I think I talked really fast. I'd like to thank the Hamilton Committee for having me here. I'm really happy to share my work with you guys, and I've had a lot of help from a lot of great people. Thank you. Any questions?
I have a question concerning the migration.
Yes.
So, are you assuming that this is just a kind of like a one-time migration event? You know, I'm looking at your arrows on your phylogeny.
Yeah. So I've made models for both sorts, continuous and different pulses of migration. Whether the pulse is happening right when the beneficial allele rises or after it's been standing for some time. But I've also worked out models where there's continuous migration happening between populations, bidirectional. Since all of these things are just based off of coalescent probabilities, you can generate lots of different sorts of more complicated models.
As a kind of a follow-up question,
Yeah.
you can generate all these models. Have you evaluated sort of the relative support for each of them?
What do you mean by relative support?
So, you know, there's a lot. You mentioned like 3 or 4 different migration models. So, is there enough power in your data to differentiate between those models?
Yeah, so I've been doing testing between runs of different simulations, and I don't know necessarily if there's power to detect between these different migration models, but there seems to be power to detect between the distinct modes. And like bonus slide, I've also been thinking about how to compare between these composite likelihoods, because they're not as straightforward as proper likelihoods. But this requires also doing simulations and taking bootstraps of those simulations. So that's something that I'm working on, is how accurately we can distinguish between these fine scale differences. Any other questions?
Thanks.
Okay, thank you.